I am Louis Gruber, a founder, CEO, and CSO of SIWA Therapeutics, Inc. I'm here to tell you about SIWA's two-pronged attack on cancer using our proprietary humanized monoclonal antibody, SIWA 318H, to clear the tumor microenvironment of both cancer cells and the senescent cells that support them. This ability to target both cancer cells and senescent cells makes 318H a comprehensive immunotherapy for all cancers, as well as for stopping cancer progression, metastasis, and relapse. We believe that 318H can do this not only without side effects, but with the ability to reverse the side effects of other cancer therapies with which our product may be co-administered without interference. 318H is a cytotoxic antibody, or immunotherapeutic, that attacks a SIWA-identified biomarker on oxidatively damaged cells. All cells with our biomarker have a combination of oxidative stress and an abnormally high level of aerobic glycolysis, the Warburg effect. Our biomarker is an oxidative side product of aerobic glycolysis. Cells with our biomarker are dysfunctional cells. Specifically, cells with our biomarker include cancer cells, senescent cells, and microbially infected cells, such as viruses, bacteria, and at least some parasites and fungi. As you will see on the next slide, we have shown that 318H binds to all three cell types, including cells infected by SARS-CoV-2 and influenza A. In part because I am not young, I began my SIWA research by looking at senescent cells, because they have a critical role in aging and aging-related diseases. Our first in vivo study, which I'll talk more about when we get to slide 7, was done in very old mice. We showed that we could reduce senescent cells and regenerate muscle tissue back to the level of young mouse controls. Next, I determined that our biomarker was not only on senescent cells, but also on cancer cells. Because cancer is a leading cause of death in the elderly, it is included as an aging-related disease by the CDC. So our second mouse in vivo study, we showed that we could statistically significantly reduce cancer metastatic lung foci in a mouse triple-negative breast cancer model while retarding tumor growth. As a result of this study and the interest of a major institute in working with us on pancreatic cancer, we are now focused on cancer, specifically pancreatic cancer. However, I do want to stress that in addition to cancer, our biomarker is associated with many other CDC aging-related diseases. For example, as a preliminary test toward a possible study in renal disease with a university group, we recently showed that our biomarker appears on, and 318H binds to, oxidatively damaged kidney cells related to chronic kidney disease. They too move to aerobic glycolysis under oxidative stress. As an indication of 318H's potentially broad effect on health span, our biomarker correlates with all Society of Consultant Pharmacists' common chronic conditions of the elderly. Because of our work on these aging-related diseases, including cancer, last year SIWA was named a key player in anti-aging drugs by the MIT Technology Review in its list of the 10 breakthrough technologies of 2020, and I was named to the Aging Analytics Agency's list of 1,000 longevity leaders for my work with SIWA in the development of 318H. This slide shows images of some in vitro binding results of our 318H or 318H mouse homologue with cells from tissues associated with cancers and other diseases. The two images to the right of this slide show 318H binding to PDX and PSN1 human pancreatic cancer cells. This was preliminary work done for us at the institute that is working with us in pancreatic cancer to show that 318H targeted these human cancer cells. Also included are disease-associated glioblastoma cells, Alzheimer's, senescent vascular microglia, and human osteoarthritic chondrocytes. Among results not shown are demonstrations of binding to kidney podocytes made senescent and to alpha-synuclein-associated cells from patients afflicted with Parkinson's disease. As I mentioned previously, oxidative stress and aerobic glycolysis, and thus our biomarker, correlate to the leading causes of death for the elderly as identified by the CDC, and to all of the common chronic conditions of the elderly listed by the Society of Consultant Pharmacists. 318H targets a biomarker for oxidative stress on cells that are using aerobic glycolysis. Oxidative stress is, of course, a major factor in cancer, including cancer resulting from oncogenic viral infection. In addition, all virally infected and senescent cells exhibit elevated glycolysis and oxidative stress, conditions which form our biomarker. Also, senescent cells are particularly susceptible to viral infection. The common metabolic profile of the three types of cells with our biomarker allows them to be targeted by 318H, which is directed to a common obligate metabolic side product, our biomarker. Perhaps the hardest task has been to narrow our focus to an initial disease to pursue. We were helped in that, in part, we were approached by a major institute to work with it in pancreatic cancer. It fit our parameters, the most important of which that it be a life-threatening disease with unmet medical need, thus eligible for FDA fast-tracking. Additionally, pancreatic cancer would allow us to possibly look at cachexia and desmoplasia as supported by our ability to target senescent cells. And as I will discuss in slide 7, we do have in vivo results relating to sarcopenia. We expect to file our first IND in late 2021, jointly with the Institute in Pancreatic Cancer and possibly in cachexia. We expect that a phase 1 basket trial in solid tumors, which could be done with the Institute, which will begin thereafter. As disorders of the GI tract often result in renal injury, we are working with another institute on a specific renal application as a possible second indication. According to researchers at Johns Hopkins University, more than 50% of seniors over the age of 75 are believed to have kidney disease, so it too fits our parameters. We plan to push further the development of 318H as a pharmaceutical in areas that we are not working in by way of licensing. We have also begun the process of moving from small batch production of 318H to a facility satisfying the FDA's current good manufacturing practices, CGMP, for large-scale production. Slides 7 and 8 show in vivo results with a 318H mouse homologue, 318M. For our 318M in vivo work, we first showed, in vivo in mice, that it targeted and removed senescent cells and promoted muscle regeneration. This slide shows the results of injecting 318M, which targets our biomarker, into very old, naturally aged mice over 20 months old, at an age at which untreated old mice exhibited substantial muscle wasting as measured by their gastrocnemius muscle mass, alongside comparisons with untreated old mice and alongside young mouse a few months old. 318M was administered twice daily, once a week for 3 weeks, and it reduced senescent cells to the level of the young mouse controls, seen in the graph on the left. As it had been reported that senescent cells inhibit adult stem cells from regenerating tissue, we showed that removing senescent cells by 318H allowed 318M allowed muscle mass to be restored to the level of young mouse controls, as shown in the graph on the right. No adverse effects or regressions were observed during a 9-week post-treatment, treatment-free period. Importantly, given the success of treating aging-associated muscle wasting by using 318M to remove senescent cells, we expect to be able to apply 318H to a variety of wasting diseases including sarcopenia, frailty, and cancer cachexia, particularly associated with pancreatic cancer. All of these diseases have been associated with secretion of tumor necrosis factor, As a suggestion of this applicability, we have shown that, in vitro, the 318H mouse homolog binds to tumor necrosis factors secreting monocytes, which are themselves associated with an aging-related monocyte subpopulation. Regarding cachexia, pancreatic cancer cells exhibiting high glycolysis trigger cachexia in animals in which the cells are implanted. 318H targets oxidatively stressed cells exhibiting elevated glycolysis. Slide 8. The 318M mouse homolog statistically significantly reduced metastasis in a 41 triple-negative breast cancer model, consistently with tumor reduction. We have seen no side effects of treatment in vivo studies in mice using the 318M homolog. Regarding 318H, we saw no side effects of treatment in non-human primates in toxicology and pharmacokinetic studies with 318H by Charles River Laboratories. Importantly, we have a two-pronged attack on cancer, hitting both cancer and senescent cells. When we began our in vivo mouse studies in pancreatic cancer, we already knew that 318H would bind to both PDX and PSN1 pancreatic cancer cells. We first confirmed that the cancer would grow in our humanized mouse model in a small sample of immune-humanized mice. We observed what we believe was a rather remarkable and very interesting response in one of the animals that were implanted with a PDX tumor. These mice were being tested only for tumor growth and therefore without any treatment. One mouse, number 9, was bearing a large tumor and was exhibiting drastic weight loss suggestive of cachexia. The animal was nearing a weight loss close to 20%, which would have required it to be euthanized according to protocol. I asked if, rather than euthanizing it, it would be possible to give that mouse, number 9, an injection with 100 micrograms, which amounts to 5 milligrams per kilogram, of 318H. As this was an amount that had been deemed safe in our tox and PK studies in non-human primates, the decision was made to do a pilot study of efficacy on 318H treatment in that mouse. Interestingly, as a result of this 318H treatment, the mouse started a rather remarkable recovery of the weight, almost to normal weight. When we last checked, mouse 9 was doing well, and there was no longer a palpable tumor. We are continuing to monitor this result, and we will be happy to share more information when available. Again, although this is an anecdotal event, we believe it is quite encouraging support for our large-scale studies and controlled studies which are in process. Charles River Labs did a pharmacokinetics and tolerability study in naive non-human primates. The study employed a single intravenous injection of 318H at 3 dose levels, 10 milligrams per kilogram, 30 milligrams per kilogram, and 100 milligrams per kilogram in naive cynomolgus monkeys ranging in age from 3 to 18 years old, including males and females. At 10 milligrams per kilogram, a dose successfully used in mouse studies, the half-life of 318H was determined to be 115 hours, or over 4 days. In all of the primates, 318H was well tolerated with no adverse effects, which is consistent with the lack of adverse effects in all of our prior in vivo studies. I want to highlight what we think are some important advantages of our 318H cancer treatment approach. By targeting our biomarker, 318H can remove not only cancer cells, but also the senescent cells that feed the primary tumor and the senescent stromal cells, and these senescent cells create an immune-suppressive environment protecting the tumor. As senescent cells are also a cause of adverse side effects from other therapies, for example by cancer or other cells made senescent by genotoxic cancer therapy, 318H's ability to remove senescent cells can retard adverse effects of other treatments. 318H also targets cancer cells that escape senescent and causes cancer relapse, resulting in an even more aggressive cancer. On the other hand, 318H does not interfere with intracellular processes. For example, the senescence and aerobic glycolysis processes. Thus, it can be co-administered with CDK4/6 inhibitors, which rely on senescence for therapy, or immuno, chemo, and radiotherapies, as well as being the primary treatment through a combination of cancer and senescent cell clearance. Considering our patents, claims have been granted in almost every jurisdiction where we've sought them, including Australia, China, Europe, Japan, Russia, South Korea, and the United States. The United States claims are not limited by medical indication. Furthermore, product claims to humanized antibodies have been obtained in Australia, Europe, Korea, and Mexico. Prosecution is continuing in many jurisdictions with an eye toward expanded patent coverage. Key personnel at SIWA include Gabriella Rossi, our CRO, and Misty Gruber, our CFO and CAO. We have a combined experience in biotech of over 75 years. I want to mention that we have focused on using contract research organizations and consultants to help to get us where we are today. It has been a very efficient means of development for us, as we have had the ability to draw on substantial expertise and equipment at essentially an hourly rate. We plan to move some consultants inside of SIWA with our next funding. Dr. Rossi adds to the clinical development experience of our team, having been a postdoc at the NIH and National Cancer Institute before moving into the private company side, where she oversaw both clinical trials and manufacturing. Her work has included clinical trials development and immunological monitoring of patients in clinical trials, as well as process development for the manufacture of cGMP biological materials for clinical trials development. We are currently looking for a Series B round of financing to get us to an IND filing, which includes a move to cGMP production of 318H. That work will be done by an entity that has been working with us and the entity that does our small batch production since we began producing 318H. Although I will not address slides 14 and 15 specifically, information on our capitalization and some comparable company valuations are included as slides 14 and 15. Our contact information is listed here. Thank you. Please let me know if you would like a copy of our slide deck.